Hi, you're listening to a conversation with Professor Henry Greely, a distinguished professor of law at Stanford University. Professor Greely is a leading expert on how biomedical technology, especially reproductive technology, relates to law and society. He's published 2 books on the subject, The End of Sex and the Future of Human Reproduction in 2016, and CRISPR People, The Science and Ethics of Editing Humans in 2021. In our conversation, we start off discussing Professor Greely's stance on using germline genomic engineering to enhance human capabilities. We then discuss several topics, including the merits of germline genetic engineering versus somatic gene therapy, and how effective or ineffective germline engineering is likely to be. Unfortunately, I messed up my screen recording setup, so my face is awkwardly superimposed over Professor Greely's. I want to apologize to Professor Greely and to the viewers for that mistake, but I think the conversation speaks for itself. Without further ado, my conversation with Professor Henry Greely. I've read some of your work, partly in preparation for our chat, and my general impression is, correct me if this is wrong, but my sort of summary or understanding would be you are definitely against a total moratorium. You're in favor of general regulation in terms of prohibiting unsafe uses, and you are. But I guess what I'm not clear on what your actual stance is on enhancement. Besides, it's not feasible soon.
So, two things. I think the general distrust and dislike of enhancement is dumb. I mean, all we've done as a species since coming down from the trees is enhance ourselves.
Mm-hmm.
That's sort of our goal, to make our lives easier and simpler, and often we do that through enhancing various aspects of ourselves. And you and I are not having this conversation face to face, even though we're having this conversation face to face. So this is an enhancing technology. I think it is morally irrelevant. As a first line matter, morally irrelevant whether the enhancements are internal to our bodies or are external machines. And yet, many people are much more freaked out by the former than they are by the latter. And I think that's kind of silly. So I'm not opposed to enhancement. I do think enhancement brings with it serious questions about equity, access, distribution, and so on, that might be strong enough in some circumstances to lead me to support limiting enhancement, but what I'd rather do is expand access to enhancement. I also think that enhancement is a very sexy topic that lots of people get really excited about. Some people get really excited about it, about using it to make money. And some people get really excited about it as an idea. And I think in both cases, expectations about what we can do often outrun any plausible reality. Right now, with respect to genetic editing, I think the possibilities for significant enhancement, doubly significant enhancement, a significant amount of enhancement in something that's important, that is significant, are quite low. And that may always be low. It may get better. It surely will get somewhat better. I think it is an open empirical question whether in 50 years we will know enough about human genetics to be able to know the alleles that significantly enhance important behavioral traits. Maybe we will, maybe we won't. We clearly don't now. And I don't think we're going to in the future.
Yeah, I guess.
So
Well, I wanted to.
that's kind of, and then my general perspective overall on genome editing, I think there are three basic groups. There are people who say, never, 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 we don't know enough to mess with our futures. There are people who say, let it rip. And there are people who say, yeah, but only for serious diseases. Um, I'm, and I think all of those are within the larger universe of, which includes almost everybody responsible, only when it's been proven safe. So if it's proven safe, there's some who say let it rip, there's some who say no, you should never do it at all, and there's some who say you should only do it for serious diseases. I kind of take a position that makes everybody unhappy with me because I think the we should never do it is wrong. I don't think there's any good argument for that. I think the between let it rip or only for disease, because I don't think enhancement is practicable at the genetic editing level at this point. And if you count skin color or eye color in a broad sense of dark versus light as enhancement, okay, we could do that. But so what? I think that there are very few situations currently where, well, there are very few situations where it seems to me that within the time frame that would be required to prove that genome editing was safe, there won't be easier alternatives for most of the uses that people have in mind, most of the treatment uses or disease uses people have in mind. So my ultimate position on it is I don't think it's really important one way or the other. I don't think it's a red line that should never be crossed. I think that's dumb. But I also don't think there's any great need for it that can't be done otherwise. Something I don't usually go into in detail, if it's around long enough and the safety is good enough and people and the societies are comfortable with it, because I don't think you can get too far ahead of your culture, then yeah, maybe it would be a good idea to take a bunch of things. Oh, and our genetics knowledge got good enough to take a bunch of things out of our genome, if only to make it something that would make a lasting difference, not something you have to do each generation over again. So let me back up. I think that somatic cell gene therapy is going to take care of a lot of the monogenic diseases.
Mm-hmm.
In which case you don't really need to do the germline, except if you do the germline, then you fix it forever or until a new mutation.
Right.
If you do somatic, then you've got to redo it every generation. And I can see that. I can see not having to redo it every generation as an advantage, although not a huge advantage.
It's also probably it's easier to develop the somatic therapy because you have faster feedback, but it is probably in an absolute sense more challenging because you have to deliver the therapy to billions or trillions of cells, whereas with the germline,
That's certainly true. And actually, by the way, Zvi.
yeah.
So that's right, but is that a plus or is that a minus? Because if you're delivering it to all sorts of cells, then maybe you're delivering it to cells where it's not necessarily a good thing. The big issue with that right now is with somatic cell, you want to try to make sure you don't deliver it to gametes or the cells that give rise to gametes because of fear of multi-generational effects.
Mm-hmm.
But let's say you've got a change that does great things for the liver, but it greatly increases a risk in the brain.
Mm-hmm.
The post-birth or the targeted somatic interventions have an advantage in that case.
Right.
Do I have any examples of it? No. Could it happen? Sure.
I mean, I do. Yeah, I guess it would be interesting. You're surely aware of like antagonistic pleiotropy between cancer and aging, specifically
Yep.
the hypothesis that the I guess what is it you have a more vigorous if you have a more vigorous youth, then your cells have been metabolizing faster and accumulate more damage, and then you get more cancer or something.
That's
Something like
yes,
that.
I have heard that hypothesis, which is plausible.
So if you
I
could target,
I
depending on the organ, you care more about the cancer versus the performance.
right,
Maybe. Yeah.
right. Or what would be another good example? Uh, well, yeah. I mean, I guess the cancer ones are good. You've got a you want better no immune system. So
Mm.
stronger immune system sounds like a good thing
Mm-hmm.
until you run into autoimmune diseases.
Mm-hmm.
So in genetics, as in almost everything else, too much of a good thing can be a bad thing.
Yeah.
And which is not at all to say that what evolution has and chance has gifted us with is the best of all possible worlds. I don't think there's any, evolution doesn't care if it's the best. It just cares if it's good enough to keep reproducing. It doesn't care at all, to be
Mm-hmm.
honest, but
Right.
it's not an optimizer. It's a satisficer. Um,
I mean, I guess like to me the sort of the general reason that germline is exciting in terms of disease is just that there's going to be a lot of um, damage basically to the genome, like purely deleterious, more or less purely deleterious genetic variants, and those ones you generally would just want to um, edit edit away.
yeah, but well, so what do you mean there's going to be a lot of damage to the genome?
Well, there's going to be a bunch of genetic variation that has pluses and minuses, or has historically had pluses and minuses, but then there's also going to be a bunch of genetic variants that are just bad. Basically, they're there
Are
because
you
of
thinking? Okay, are you thinking about new mutations or?
yeah. So yeah, de novo mutations or generally rare mutations.
Yeah, okay.
Um,
Yeah, I mean, almost all genetic variations make no discernible difference in anything, and a few of them are bad, and an even fewer of them are good. But at least in terms of our health, I think the role played by mitotic damage, by somatic mutations, is for most of us bigger than the role played by inherited variation. And cancer is almost, cancer is, I believe, mainly, I think this is not a, a, a controversial thing to say, most cancer is probably the result of bad luck and mistakes during cell division. Yeah, radiation can increase that and carcinogens can increase that, but most of it is, when the cell divided and A turned into a C and bad shit happened. Or there was a two base pair deletion and bad stuff happened. So yes, there are bad mutations, but an awful lot of the bad mutations are mutations that happen during your lifetime, no matter
yeah.
what
So
germline you start out.
I mean, this matches my understanding, and yet I also suspect you can have a pretty large effect through germline editing because there's still like a good chunk of heritability. I think even with cancer, the numbers I've seen for various common cancers are kind of like 10%, 20%, 30% heritable, which doesn't translate into we can edit that now, certainly.
Right,
But
right. That's that. I would think a little bit lower than that, but yeah, that's not an unreasonable range. Um, it is sort of frustrating. You look at that for, say, breast cancer, but on the other hand, you look at BRCA1 and BRCA2, and the number of women, the percentage of women who get breast cancer who have germline mutations in BRCA1 or BRCA2 that we know or suspect to be pathogenic is a very small percentage. It's like 3 or 4%, 5% of the women who actually get breast cancer. And there's more out there. Will we find it? Maybe. Are you familiar with the Vischer paper in January in Nature?
I've seen it. I didn't read it carefully, but yeah.
Yeah, it's one you might want to read carefully. It's one I disagree with, but
Okay.
actually was co-author of a three-person response. Um, but it argues that you could massively reduce the incidence of a variety of common diseases by making 10 to 20 edits, germline edits in babies' genomes, which would be assuming that you knew how to safely and effectively make 10 to 20 edits in a germline genome might be attractive. Um, I recruited a couple of geneticists to write the response with me, and they're using GWAS, so they're using, SNPs, and they don't really know what the causation is, and basically the science isn't there right now for us to be able to say, yes, if you make these following 10 specific edits, you will have a 80% reduction in asthma. Might be eventually, but we're nowhere close to it yet. And it might not be eventually.
I mean, I
And
I guess basically, so I'm not a geneticist by any account, and I'm not even well read because I've just been focusing on other things. Um, but my kind of sort of lay understanding is that, uh, or my maybe this is just an intuition and it can just be brushed off as an intuition, but I would think that generally just like biological systems are sort of complicated and have many routes by which they can accumulate damage and also therefore by which they can be kind of like fixed or tuned up. So I would just generally expect there to be lots of polygenic influence on many traits, like disease,
yeah,
lots of diseases.
yeah.
And so that kind of suggests to me that we should expect there to be lots of causal levers on these disease traits. What do you think about
So
it?
I think your intuition there is correct, although a corollary of that is because there are so many different pathways, there are also a lot of workarounds. One of the more interesting things of doing whole genome sequencing is finding perfectly healthy people who look like they should be dead, who seem to have mutations in life essential genes, and yet they're okay.
Mm-hmm.
What's going on? Well, there's some alternative route that exists and that they're using. Um, so I think that's right. I think there are lots of polygenic influences on things, but figuring out which combinations of alleles lead to which results is really hard. And we're nowhere, I think, close to being able to do that in maybe in any cases. I mean, the number of polygenic, strong polygenic disease risks we know about is very, very small. Well, where we can say this somebody who has this version of this allele on this gene and that allele on that gene has a 90% chance of getting this disease. Um, we know a lot. We know about 7,000 or more diseases where we can say if you've got this allele on this gene, you're got 90% chance of getting the disease. The combined ones we don't understand very well. We may never understand very well. Conditions, diseases, traits are affected by gene sequence, sometimes really powerfully, are affected by environment, sometimes really powerfully, and are affected by chance, sometimes really powerfully. And the interactions of those are going to be hard to tease out. For some things like Huntington disease, it looks like environment and chance play basically no role. But those diseases, those powerfully monogenic diseases, seem to be quite rare. And the polygenic diseases, yes, it's undoubtedly the case. And this is what the whole PRS stuff is about. It's looking at GWAS and SNPs and saying, hey, we see that when you've got this SNP, which gives us a slightly higher chance of thinking you've got a particular sequence somewhere in the area of that SNP, you are at higher risk for this. That's interesting. I think it's mainly interesting as a lead to genes to then explore actually regions of the genome, because the SNP doesn't give you the gene, it just gives you a neighborhood. Regions of the genome to explore more carefully, but it doesn't. So there are, say, 20 hits, 40 hits on the SNPs for height. But that doesn't mean that any of those is necessarily important for height, or, and this is a big problem with PRS, or that their effects are additive. So one simple model, let's say you've got 10 different genes that you know are significantly involved in colon cancer. But none of them is really powerfully involved. That's actually, colon cancer is a bad example because there are some genes that are powerfully involved, but pretend that's not true. So you got a disease, 10 different genes, they seem to be involved. Each one seems to add 5% to somebody's lifetime risk. It is, and by I'm using gene, I should say allele, each particular variation or allele. Particular variations or alleles add 5% to your risk. The tempting thing to do, which PRS generally does, is add them all together and say if we could modify all 10 of these, we can take care of 50% of the risk. But there's no reason to think that's true. The effects could have no effect, could be countervailing. Having two bad things could actually weirdly turn out to be good. The one thing I've learned most in 35, 40 years of deep dives into human biology is that biology is just weird.
Right.
It's not logical. It's not designed, It's evolved and weird things happen. So
I,
my
I,
main concern
I,
here is I think we don't know enough. And maybe we'll know enough at some point, but I don't think it'll be soon. And know enough, one other thing, know enough about what can make people better, what combination of alleles can reduce the risk of, say, schizophrenia. That'd be a good thing. I hate schizophrenia, worst disease in the world. And I think, and they're clearly leaving aside the issue that schizophrenia probably isn't one disease, but a bunch of different things that share a name. If you could do gene editing to reduce schizophrenia risk, that'd be a great thing. I don't, right now, we're nowhere close to understanding, we haven't found anything that has any single allele that has a powerful effect. But the other problem is, maybe if you reduce schizophrenia risk, unknowingly, you're greatly increasing the risk of type 1 diabetes or brain cancer or the heartbreak of psoriasis or something else.
well, so okay, so there's two things I want to ask you about. One thing is,
Yeah.
I mean, I sort of hear the debate a little bit from afar about causality, and it's confusing to me because, or not just causality. So there's the question of causality, and then you also importantly brought up the question of additivity. Like if we edited or selected for 10 or 20 or 50 variants, alleles, are the effects adding up, or are they completely, maybe they have the opposite effect, or they just hit quickly diminishing returns.
Right.
And I'm always like, I feel like the obvious thing to expect is that they add up, you get diminishing returns, but the diminishing returns are not that quickly diminishing. I feel like that's just sort of the obvious thing to expect, and I don't know.
But so I think my main concern about that is that the obvious thing to expect is so often not true in biology.
Mm-hmm.
It's true in many of our normal activities of life, but in biology, it's not only is it not necessarily true, but it's often not true.
Here, let me ask you this.
Oh,
What sort of
and let me give you one kind of example.
cool.
So maybe you get no additivity at all because there's a particular cascade of there's a network of a biochemical network that when disrupted causes the disease. And you've got 10 different spots, but each one disrupts a different crucial part of that cascade or network. And so, is that what I want to say? That, well, no, maybe you've got 10 independent paths. Anyway, it's possible that they're all in the same network and it makes a big difference, or that they're all in different networks, and so you, you don't get any additive.
Yeah, I could. Well,
Forget it.
I could try to give
But
an example.
additivity is complicated.
Yeah, so I mean, an example where additivity would fail catastrophic, or a real simple example where additivity would fail catastrophically would be height, where we do know lots and lots of genetic variants associated with height. Imagine that
Mm-hmm.
they do add up in a straightforward way, so you can make someone who's 7 feet tall, 8 feet tall, 9 feet tall. But it's not going to be additive forever, because if you make someone who's 9 feet tall, he's just going to die, um, and
I
he won't be
think the
very
world's
tall because he'll
tallest
be dead.
person, yeah, I think tallest person known did hit a little over 9 feet. He
Oh,
died in the
I thought it
Swiss.
was 8 foot 11. It's Robert Wadlow.
Is this the Irish giant, or? And if it was 8'11", I might just have 9 feet stuck in my head because it's close. It's
Um,
around 9 feet. But
he's known as
anyway,
the Alton Giant, or the Giant of Illinois, and he was 8 feet
uh,
11 inches.
okay. I forget how tall the Irish giant was. This is an early 19th century guy who was also really tall. But yeah, so additivity will be self-limiting at some point, if in fact things are additive.
Mm-hmm.
But of course, we don't know whether he was so tall because of 10 different variants that all lined up the right/wrong way, or because of one variant that, grossly pumped up the human growth factor or something else.
Mm-hmm.
And ultimately, so assume for a second you're interested in 10 variants, 10 genes, and each one has, no, 20 genes, and each one has only two variants, and each variant is distributed 50-50. So a very artificial thing. You got 2 to the 20th, which is roughly a million.
Mm-hmm.
So you've got a million different combinations. In order to tease out the effects of each of those, you're going to have a really good database that has a lot of data on a lot of people. And you won't just want 1 million people with a likelihood of getting one person with each combination. You'll want 100 people with each combination. You'll want to follow them throughout their lifespan. And you would like it if they had roughly similar environments, although, part of you would like it if they had different environments. I mean, quite quickly you get to wanting to have a sample that's bigger than the total number of humans who ever lived.
I'm not sure I buy this. I mean, if we're talking about fairly common variants, it's true that if you're worried about these interactions between 5 or 10, or you gave the number 20 genes, yeah, you're never gonna see every combination. But uh, I I don't know what to. Okay, I don't know what to think. Well, I don't like it. I feel like we should if there's like all these like deadly like 10 gene combinations where like any set of 5 of them is perfectly fine, but then like when you get these 10, I mean, do people just like die randomly if like for no known reason?
Um, so sometimes people drop dead for reasons that don't get explained or understood.
Mm-hmm.
And often that's because nobody's done an autopsy or nobody's cared enough to actually look that hard. But sometimes it, there will almost always be something a doctor can say, yes, they died because their aorta burst, or they died because um, their brain neurons started dissolving
Mm-hmm.
without necessarily knowing why either of those things happened. So I remember one of my science fiction favorite old science fiction authors had a line somewhere. Um, he died of heart failure, and the guy who's being told this, who suspected that the man in question was killed by the police, thinks, "Huh." But ultimately, as my biology teacher said, since we define death by the heart not beating, everybody dies of heart failure.
Right.
So you can always say he died because his heart and lungs weren't working anymore,
Right.
or because his brain wasn't working anymore. But do we fully understand it? But I see your point. But maybe if you've got the wrong lineup of alleles, your embryo never implants or never thrives,
Mm-hmm.
or maybe it's fetal lethal. Um, what you what you'd really love to do is make 1,000 clones of each set of variations that you think are important, have them be born, and watch them for 80 years.
Uh-huh.
Um, we're not going to do that, but that doesn't mean. So I'm not saying, I'm not putting this forward as this is always going to happen, this is always going to be the case, but that there is a realistic chance that it is the case, and people who get enamored of genetics often assume that it is more powerful and more predictive than it actually has turned out to be.
Mm-hmm.
If you go back, I keep saying I'm going to write this. I got to write this. I've had an idea and a title. The title is "It's More Complicated Than We Thought."
Mm-hmm.
If you go back to the year 2000, we get the first draft of the whole human genome. We get human embryonic stem cells. We get fMRI. 3 different advances in 3 different areas, and each one has reasonable people saying, "Wow, within 20 years we're going to be able to fix everything." And we're now 25 years on, and they're good technologies. They've helped some things. They've actually helped some people, but it's a lot more complicated than we thought.
I think
And
the big thing to me is
the
just
universe does not have to be set up in a way that lets us understand it.
that's right. That's right. I think the big thing to me is just, I mean, you're the expert. You're an expert on what I'm about to bring up, but
But my last biology class was in 10th grade, so I'm
right.
not
But
a geneticist either.
on the social aspect, it seems to me like we're very much like only a half of a foot in the door and 1.5 feet out of the door with regards to society actually wanting to, or invest in the possibility of this technology. And so we haven't really tried to figure out how it could work. Yeah.
Yes and no. I mean, almost every kid in the U.S. gets a genetic test shortly after birth for, in most states, about 50 different conditions.
Mm.
That's a pretty big foot in the door. Almost all of IVF now in the US, almost all, maybe too much, 80% or so involves pre-implantation genetic testing.
Mm-hmm.
That's now granted, IVF is only 3% of births, and that's a select sample of people who choose IVF, but I'd say that's more than a foot in the door. Um, I do think that there is a strong and, to me, irrational concern about germline, but there are also certainly there are some real rational concerns about germline in terms of safety at this point. I mean, one of the great things about doing somatic, if you're able to target the edit and you put uh, CRISPR into a kidney and with a gene with a replacement that improves kidney function or saves the kidney, it doesn't matter that it has also there was an off-target effect that would kill brain cells because it's not going into brain cells, or there's an off-target effect that would mean that an embryo could not progress from week 6 to week 7 because it's not doing that sort of development. So one of the advantages of targeted somatic therapy is if there are errors, most of them will not be relevant to the particular, many of them would not be relevant to the particular organ you're trying to affect. Whereas if you do embryo editing, you're seeing it, you're affecting everything. Yeah, I mean, which is the advantage, right? You affect all the cells at once,
Mhm.
which is the disadvantage. You affect all the cells at once.
Um, a thing I wanted to ask you earlier was regarding the additivity.
Yeah.
Um, what is there something that you think we might, some sort of study we might be able to do that would convince you, oh, actually, maybe these things are fairly additive and we could have fairly large effects with multigenic editing?
Yeah. Um, you need something like the UK Biobank, lots and lots of people. You need them to be whole genome sequenced. I think the UK Biobank has about a quarter of its people whole genome sequenced at this point, but they're aiming to get more. You need really good health information, which the UK Biobank is probably the best thing we can do. I don't know whether the NHS's regular electronic health record, just how good it is. Um, but it's going to be better than the mishmash of crappy electronic health records we have in the US, which are mutually incomprehensible and designed mainly to maximize billing rather than to maximize medical information. And you would say, okay, I want to see, and you need some pretty common variants because otherwise the numbers get really small really fast. So if we keep with my, if we do 10 variants and we keep with 50-50, you got basically two alleles and each one is 50% represented. So you would get each combination one time in 2 to the 10th, which is basically 1 in 1,000. So in a million-person biobank, you'd have 1,000 people. And with full health information on 1,000 people, yeah, it would be nice to see, and it would be, let's say it's about colon cancer, fairly common disease. 2 or 3% of the population will get colon cancer eventually. And you might want to see this just nice linear graph. With each additional bad variation, you get a proportionate increase. Yeah, that would be a really convincing piece.
I did
Now,
actually.
I would also,
I tried to find studies like that, if I understand what you're saying, and I couldn't immediately
yeah,
find any. But if you know geneticists who want to run such a study, I'd love to chat with them.
so read the Visscher's piece closely
Okay.
and look to see who he cites. Now, and read our piece. It's Carmi, Greely, and Mitchell. It's in the same issue, January 9th issue of this year's Nature.
Okay.
Uh, because Carmi and Shai Carmi and Kevin Mitchell are both really good geneticists, and they will poke holes in the Visscher's analysis, but um, they are trying to argue that other part of the problem is they're using SNPs rather than whole genome sequence. So SNPs are not very useful markers, or they're not very precise. You know about SNPs, right?
Yeah, so you're gonna you first of all, the causal things might be rare variants that aren't even showing up in SNPs, and even if it were SNPs, you're gonna usually be very uncertain between a block of 5 or 10 or 50 adjacent SNPs as
Y-
to
y-
which one is causal.
Yeah, but I think the fundamental issue with SNPs is most SNPs aren't in are likely to have no importance on their own at all. Most of them are not going to be in coding regions or regulatory regions because most of the DNA, most of your genome isn't in coding or regulatory regions.
Hmm.
It's in what careless people call junk DNA and what careful people call quote junk close quote DNA. So most of the SNPs are meaningless, but the SNP is more, a particular SNP is statistically in a particular population more likely to be associated with meaningful stuff within a couple million base pairs in either direction. And so it's something in that couple million base pairs that is presumably having the effect, but you don't know what it is. If you had whole genome sequencing and you could then figure out what's actually having the effect, that'd be nice. The problem is SNPs are, probably at a big research lab, they're probably $50 or less each, and whole genome sequencing is still a grand or more.
Hmm.
800, 1,000, somewhere in there. And
That's not
depends
what
on
I've
how
heard.
good, it depends on how good quality you want. What have you
Yeah.
heard?
Um, Last time I looked it up, it was, yeah, I don't remember the coverage, but the number was more like $250 or $300.
So Illumina has long had a lock on human sequencing, like 90% of the market, and they've been able to keep the price fairly high. Cost and price are very different things. And I think that there probably are people who can deliver a whole genome sequence for $250. I don't know that anybody is doing that at scale. And then the other issue you've got to ask is what quality are you getting for that? And I don't know, but I do think it should ultimately be $10.
Well,
I think
so
Illumina's de facto monopoly has really been a bad thing.
so Nebula's, I'm trying. Last time I looked up so Nebula is George Church's company,
Yeah, yeah.
and
Good
it's
old George.
it's 30x coverage. Last time I looked it up, it was $250, but maybe that was some one-time offer.
And how many of them are they doing? How widely accepted is it? George is an amazingly creative guy and a really interesting guy, and I love talking to him. And he's founded probably 500 companies, a few of which have actually done something useful.
Hmm.
But in the biotech space, if you hit on 1 out of 50 startups, you're doing really well.
Hmm.
So
Yeah, I'm not sure. I'm
I
not finding
am,
how many
yeah,
they have
I
done.
am, I am suspicious of outliers.
Hmm.
But I do think, and I think that if Illumina had had solid competition, we would have seen this 5 or 10 years ago. And I think now Oxford Nanopore and there are people who are doing completely different routes rather than the shotgun sequencing that Illumina is relied upon. I think we should see the cost of of good whole genome sequencing come down a lot fairly soon. And actually, I think even the $1,000 that I think is low for clinical sequences because they want a higher level of coverage and accuracy with clinical sequences than research sequences. But yeah, whatever it is, SNPs are a shitload cheaper than whole genome sequences. So we got a lot more SNPs and a lot fewer whole genome sequences. But
Yeah,
the
something like an
whole
order of
genome,
magnitude more
yeah,
genome-wide SNP arrays. Yeah.
I would say probably more than that.
Well, my count was something like, I manually counted. I got something like 2 million whole genomes, but people suggested it's more like 5 million whole genomes, or
Yeah,
10 even. And then the SNP arrays, you've got those big ancestry companies like MyHeritage,
yeah.
and they've got over 50 million SNP arrays. you're
Yeah,
saying,
that sounds. I think the genealogical companies between them probably have over 50 million just in themselves.
yeah,
And then there's a whole bunch of research that use SNP arrays and um, not been used for forensic purposes much, but I'd be surprised if we aren't well over 100 million SNP arrays, SNP results. If we don't have SNP array results for over 100 million people,
you'd be surprised if we don't. Okay,
I'd be surprised if we don't, which is nice.
yeah.
It's just not that nice.
Um, do you think that the data being uh, generally not accessible is mainly privacy or mainly because people don't want to use it for predicting traits or what?
So, what do you mean by the data being generally not available?
Well, it's available for some research. I don't know the details, but I generally hear people having difficulty doing um, genetics
So one
research.
reason that it's one reason it's unavailable is that the companies that hold it don't want to share it because they want to get all the profits from it,
Mhm.
or they'll only share it if you pay them a lot of money for it. Another reason is people are very worried about genetic privacy, and those worries both have legal implications in terms of laws about genetic privacy. They have implications in terms of the consent forms or uh, terms of agreement that people who've given their DNA to a company have signed, and they have public relations consequences. So if you're a company or a government entity sitting on a lot of data, you're nervous about privacy. That data is, if you're a company, it's an asset that you'd like to get value for. And if you're not a company, and whether or not you're a company, it's a potential legal and public relations liability in terms of privacy. I think people are somewhat, well, I think people vastly overestimate the importance of their genetic data, but they do. And part of that is because for 30 years we've been telling them that their genome is them.
Which is a weird thing to say. It's very biodeterminist.
Yeah, but it's a good attention getter.
Mm.
Whether you're a scientist or whether you're a headline writer or whether whatever you are, an entrepreneur. And after all, in our most of our society is built on hype.
But okay, so given given that there's a lot of hype and it's hard to direct attention um, according to actually good possibilities, um, well, because I don't know if you even would I don't know if you would even say this, but I think you should say this that there are pretend I think you've said this, but you always put a lot of caveats that there just are very large potential benefits to this technology.
Okay, so which technology?
Germline genetic engineering.
Germline?
Yeah.
Okay. So I think there are potential benefits. I think many of those potential benefits could be realizable through, and may end up being realizable through methods that are through other methods that are currently better proven or more plausible, less likely to be dangerous, although germline editing might, I hope will turn out to be doable in ways that aren't dangerous. But there are alternatives that make germline editing not all that attractive. And germline editing does have a public acceptance, public reaction problem that things like somatic editing does not. And so, is the fact that the public is freaked out by it a good moral reason not to do it? No. Is it a good practical reason not to do it? Yeah, maybe. Especially if you've got good alternatives that the public isn't freaked out by.
Yeah, I guess
I'm
the
not a philosopher. I'm not an ethicist. I'm a law professor,
right.
and so the real world is what I worry about.
I think that like one of the really big important things to me is cognitive enhancement. So I mean, basically making it possible for parents to make their kids much smarter than
Yeah.
they might
Good
otherwise be.
news is we can do that. We can vaccinate them and protect them from diseases. We can feed them well. We can read to them. We can care for them. We can do all sorts of things to make our kids smarter. To literally make our kids smarter.
Hmm.
In genetics, we know almost nothing about genetic variations that improve whatever intelligence is. We know a shitload of genetic variations that powerfully affect intelligence by making it very low.
Well, I
But the fact that phenylketonuria gives you low intelligence doesn't mean that giving you an extra copy of the phenylalanine to tyrosine gene will make you smarter.
right. Well, but I don't know if you may just not be up to date, or maybe I'm not understanding the implications of the research. Um, there are polygenic scores for intelligence that
There
they
are.
explain
Although most of them tend to be more educational attainment than intelligence, but
that there are, yeah,
because
there's education.
that's easier to measure.
Right. But I think that there are actually, yeah, I don't know the details of the methodology, but I think they at least claim to be more direct
Yeah,
predictors for
there
cognitive
are.
ability, and they even get up to, like, I've definitely seen over 5%. I think the biggest number I've seen is 9.7%.
Yeah, and I don't believe them.
You don't believe them? Okay.
I don't believe them. I mean, I believe that they did the work. I believe that their equations led to those numbers. But this goes to my deep skepticism about polygenic risk scores. And, I believe that you could probably get, a couple of IQ points.
Well, is it mainly
Big deal.
is your skepticism there with the polygen? We don't need to. We're not going to be able to hash this out because I don't have a command of the facts. But I would just like to understand your general
You
sense. Like,
also don't.
is it
The
mainly?
part of my reaction is I've got 35 years of watching people greatly overestimate the effects of biology.
Mhm.
So it doesn't mean that, the fact that they've been wrong for 35 years doesn't mean they'll be wrong this time. And sometimes they're right. But um, I take everything with a grain of salt, particularly the outliers, because everything in our society tends to push toward publicity for attention to rewards for dramatic stuff. And so what you read about, what gets publicized, what gets published is the dramatic stuff, most of which turns out over time not to be right. Not because anybody was committing scientific malpractice, but there's just a lot of variation in experimental results.
So you're not going to get an argument from me on that point, but the issue for me here is, so I had asked, I had wanted to ask, like, aren't there potentially very large benefits to germline engineering? And then you had said, well, and I brought up intelligence or cognitive ability, and you said, well, we can't affect it. And then we're talking about whether the current polygenic scores are capturing enough causal variance and whether it's additive and so on. But my question is more, would it be feasible if we tried really hard? Not is it feasible right now?
Yeah, so and my honest answer is probably to some extent, but that's only probably. It may be that we will never understand it enough to be able to do much. My guess is, and I've written this, that in 20 or 30 years, we may be able to look at an embryo's whole genome and say, this has a 13% chance of being in the top decile for SAT scores, which of course means it has an 87% chance of not being in the top decile. And from 10% to 13%, It's an improvement. Is it a big improvement? I mean, maybe. If it's really cheap and easy and we're confident that there's no ill effects from it, would it be worth doing? Maybe, probably, even maybe. Um, is it likely to be a big deal? I don't think so. In part because, so I started doing law and biosciences stuff with genetics, and then got into cloning and stem cell stuff, and then got into neuroscience. The enhancement things we care about, you care about intelligence. And I think most of the things we care about are behaviors. Intelligence, music ability, math ability, sports ability, personality. And all these are mediated through the brain. And the brain is about 100 orders of magnitude more complicated than the genome. There's a good book by my co-author on this Nature piece, Kevin Mitchell, called Innate. He's a neurogeneticist. He believes that genetics does play some important role in brain development. I recommend the book because he also believes it's really complicated and teasing out the role is really hard. And that ultimately behaviors are about 1/3 genetic, 1/3 environmental, and 1/3 just plain chance. And that seems about right to me. Brains are really hard.
So I mean, I certainly buy that there's always going to be like a lot of variation that we can't control. I guess the question to me is, can we push the means around?
Maybe. And even if you push the means, so you want to push the means, do you want to push the standard deviations? We know we can push the means around by things like better nutrition, better health care,
Right.
better education, better home environments. Those affect the mean. Do you know about the Flynn effect?
Certainly have heard of it. Yeah.
Yeah, I mean, I don't think anybody's got a complete explanation for why IQ goes up about 10 points every 20 years over the last 80 years of measurement. It doesn't go up because they keep renorming the test. But are we 30 IQ points smarter on average than our grandparents? I don't think so. But if you test us, it looks like we are.
Mm.
I think we're probably better at making circles with number 2 lead pencils.
Right.
Filling in circles. But, so my basic view of much of science is there's just much of biology, at least. This is just a lot of uncertainty. And and it bothers me in health and biology because it's uncertainty in a context where people, both desperate patients, but also the media, science, universities, medical school communications offices, announce breakthroughs every month. And if 1 in 20 of them actually turns out to be important, we're lucky. And we're in a culture where hype is everything. Well, hype is everywhere. But that's not to say, there aren't great things happening. So, for example, this I think is the most significant medical miracle, with the possible exception of the COVID vaccines, of the last 5 to 10 years, and nobody is paying attention to it. 90% of people with cystic fibrosis can now live normal lives because of a small molecule drug. It's actually a combination of 3 small molecule drugs. It doesn't change the DNA sequence, but it reconfigures the protein produced by the DNA sequence of the most common variants responsible for cystic fibrosis in a way that means you're not symptomatic. That's great. That's a medical miracle. And it's in a genetic disease. But the solution didn't have anything to do. I mean, had to do with genetics because knowing about the gene and knowing about the protein helped figure out what the small molecules should do and were doing and so on. But it's not a sequence intervention. So I'm ultimately I'm optimistic on most things. I'm most skeptical about behavioral stuff because brains are just hard. And if you even look at medicine, mental illness and neurological diseases, we've had almost no progress in the last 30 years. Multiple sclerosis is maybe the one counterexample. There's been some good successes with multiple, but they're still not cures. They're ameliorative treatments. So, but I should probably do some work.
Yep. Um,
And
y-
I am. What are you trained in? What is your background? Having said you're not a geneticist, as I am not a geneticist,
um,
I'm perfectly willing to listen to talk about genetics from people who aren't geneticists because I kind of have to.
yeah. Um,
I'm stopped from disagreeing.
I studied math at university. Um,
Where'd you go?
Undergrad was University of Chicago, and I was briefly in grad school at UC Berkeley for mathematical logic.
Yeah,
But then I left to my career has been trying to make it so that artificial general intelligence doesn't destroy the world. Uh,
that sounds like a good thing.
yeah,
Although AI is another one of those things that I think is susceptible to hype.
I agree with that. Everyone around me is like debating whether we're all gonna be killed by AI in 2 years or 3 years, and I'm like,
Yeah,
is it 20 years or 50 years?
or maybe even never. But if you're successful,
Right. Well, you mentioned Kevin Mitchell's book
yeah,
Innate, and then you've written the
yeah.
book CRISPR People. Do you have one or two other recommendations?
Um, well, so I don't know how comfortable you are with your genetics background. You obviously have learned quite a bit. This might not add, but it might. Carl Zimmer is one of the best science journalists around. He writes for the Times, among other things. He has a book called She Has Her Mother's Laugh,
Hmm.
which is a great look at both the science of genetics and its history, dark sides, bright sides. I think it provides a lot of good context for understanding genetics.
Cool. Thank you.
Uh, and he's a great writer, so it's a good read.
Cool.
CRISPR People, CRISPR People is a good read. It's mainly the story of the He Jiankui babies, um, but there's a lot of other stuff in there too. Um, my other book, The End of Sex, I actually think is more important, but isn't nearly as easy or good a read. And this is some proof that I can take seemingly overly strong positions, despite my skepticism. It predicts that in 30 or 40 years, and this is 10 years, 9 years ago when it came out, so 20 or 30 years, in many countries, most people will make their babies in clinics and they will do it in order to use genetic technology,
Hmm.
but not genetic editing, genetic selection.
Hmm.
So that's the other big alternative to gene editing is gene selection through pre-implantation genetic diagnosis. The technology that will make this much more feasible is, I'm still confident, is going to happen. And every morning it could be the case that I will wake up and there will be the proof of principle. It's in vitro gametogenesis,
Right.
taking fibroblasts, making them into induced pluripotent stem cells and make them into mature gametes. Been done in mice in lots of fun, complicated ways in mice, including making eggs from male cells. Um, there are 5 or 6 academic labs around the world working in that space. They're not very many because governments don't pay for it for the most part because it's politically tricky.
Hmm.
There are now several startups that are working in various sides of this. And there's a this is the two sides of the coin, and coin is a particularly good term for this point. There is a great need for it, and there is a great market for it. So there are millions of couples who desperately want to have children that are genetically theirs, and people who go through IVF, 6, 7, 8 IVF cycles at high cost, emotionally, physically, and financially. But many of them can't because they don't have gametes, or they don't have gametes that work. So that's a lot of people who are suffering, and the philosophers, so fucking many philosophers will say, well, they're not suffering. That's not it. You don't need to have genetic children. Yeah, bullshit. I mean, if
Right.
people suffer, they suffer. Um, and this is worldwide. There are millions of them, which also means there's a big market. So I think this is going to happen. If this happens, how this plays into genetic selection and what I call easy PGD, although nowadays I guess I'd call it easy PGT because the acronym has changed from diagnosis, pre-implantation genetic diagnosis to pre-implantation genetic testing, is instead of making 5 embryos or 10 embryos and selecting among those, you can make 100, make 1,000. If you got enough money, you could make 1,000,000
Hmm.
and then do whole genome sequencing on each one and pick the one that looks best. It's not as good as editing because the parents may not have the alleles you want.
Right.
And I think the killer app for this is not going to be enhancement. It's going to be the 7,000 rare genetic diseases that we understand and which are strongly inherited, strongly genetic. They're all rare, but 7,000 times rare is 1 to 2% of births. So to do this to avoid the 1 to 2% chance of having a kid who's got a really awful disease, I think that will lead parents to want to do it and will lead health insurers and government programs to want it to be done because it's a lot cheap. It's likely to be a lot cheaper than caring for a severely intellectually disabled infant for 70 years.
Right.
So that's the end of sex and the future of human reproduction. And it proves, I think, that I can take an outrageous position.
Well, thanks for the
But for
chat.
some reason,
Yeah.
PRS and genome editing, it's it's not one that I'm bought into.
Yeah, I mean, I guess I've been chewing on, and I will keep chewing on how to test or demonstrate to the skeptics uh,
Yeah,
that the benefits could be big.
and I do think if you've got something, well, read the Vischer's piece and follow some of their footnotes into some of the things they cite, but also read our piece because Kevin and Shai have some good counter arguments as well.
Cool.
But in theory, the answer is fairly clear. You get a huge biobank and database with millions of people, with whole genome sequences on all of those people, and with complete health data on all of those people over a span of several decades. The only problem is it'll take a long time, it's really expensive, and who's going to pay for it? But that, and it still wouldn't give you complete confidence, but it would give you some, I think, non-trivial answers. Of course, if you're interested in intelligence, your problem is the health data is not going to give you much about intelligence.
Right. We need phenotypes.
Yep, and asking the million people currently in the UK biobank to all take IQ tests is probably not going to work.
Probably not.
Which is
Um,
why the people who've used that biobank use educational attainment levels because those are often in there.
yeah.
Anyway,
Um,
I don't think you're crazy.
Thank you.
I think you're over optimistic.
Hmm.
I think you're young. these two things are correlated, I think. The R
Yep.
squared is positive.
Yeah.
And I'm old, which is correlated with skepticism, but doesn't mean, but I'm skeptical even of skepticism. Sometimes stuff turns out to be right.
Guess we'll see. I appreciate the
Well,
conversation a lot.
you'll see. I don't know that I'll live long enough to see. I have no immediate plans to die, but I am 72.
Yeah.
And I'm thinking 20 years is, to get good answers to this within 20 years would be impressive.
I'll try to get you those answers.
Okay. All right. Nice talking to you.
Yeah, you too.
And